Thousands of years ago, an ancient people walked the lands of the Levant, their traditions shaping civilizations, their stories passed down through generations. But what if their very DNA carried a record of that journey? A genetic imprint so distinct that it connects millions of people today, across continents, cultures, and histories. Jewish DNA is unlike any other. It's not just about ancestry. It's a living map of migration, survival, and resilience. Over the past few decades, geneticists have uncovered something remarkable. Despite thousands of years of dispersion, Jewish populations remain strikingly connected at the genetic level. So, how is that even possible? What makes Jewish DNA so unique? And what hidden secrets does it hold? Scientists have traced Jewish ancestry back to the ancient Levant, the land that now includes Israel, Palestine, Jordan, and Lebanon. Genetic studies show that today's Jewish populations share a deep connection with their ancient Israelite ancestors. This isn't just a theory. It's been confirmed through the study of Y-DNA, mitochondrial DNA, and autosomal markers. In fact, modern Jews are more genetically similar to Middle Eastern populations like Druze, Lebanese, and even Palestinians than they are to their European or North African neighbors. But here's where it gets even more interesting. Over centuries, Jewish communities were forced to move, resettle, and sometimes completely rebuild in new lands. The destruction of the first and second temples in Jerusalem led to mass migrations, spreading Jewish populations into Europe, North Africa, and the Middle East. Despite these movements, something unusual happened. Unlike most migrating groups that eventually blend into their surroundings, Jewish DNA remained largely distinct. The answer lies in a combination of endogamy, the practice of marrying within the community, and genetic bottlenecks. Endogamy helped preserve Jewish genetic markers across generations, while bottlenecks, caused by historical population reductions, created founder effects. In simple terms, a small group of ancestors contributed to a significant portion of the modern Jewish gene pool, particularly among Ashkenazi Jews. Take Ashkenazi Jews, for example. Despite making up nearly 80% of the world's Jewish population today, they can trace much of their ancestry back to a tiny group of founders. Studies have shown that nearly 40% of Ashkenazi Jews descend from just 4 maternal lineages, a genetic phenomenon that's rarely seen in other populations. This explains why certain genetic traits and even inherited diseases, like Tay-Sachs and BRCA mutations, are more common in Ashkenazi communities. But this isn't just an Ashkenazi story. Sephardi Jews, who trace their roots to Spain and Portugal, and Mizrahi Jews, who remained in the Middle East and North Africa, also show remarkable genetic continuity. Despite being geographically separated for over 1,000 years, Jewish populations across the world still share a significant portion of their DNA. So, what does this mean? It means that Jewish DNA is more than just a connection to the past. It's a genetic fingerprint that has withstood time, persecution, and migration. It tells a story of survival, adaptation, and identity. And perhaps most importantly, it challenges the idea that identity is solely defined by geography. The Jewish people, despite their dispersion, remain one of the most genetically cohesive groups in the world. And this is just the beginning. In the next sections, we'll dive deeper into the genetic markers that define Jewish ancestry, explore the mysteries of the Cohen modal haplotype, and uncover how modern science is rewriting what we know about Jewish history. For centuries, history has been written in books, carved in stone, and passed down through stories. But what if history is also written in our DNA? Hidden within the genetic code of Jewish populations lies an ancient signature, one that connects them to a shared past stretching back thousands of years. But how do scientists actually trace Jewish ancestry through DNA? And what makes this genetic connection so unique? It all starts with something called haplogroups, genetic markers that are passed down from parents to children, unchanged for generations. These haplogroups act like timestamps, allowing geneticists to trace lineages back to their origins. Among Jewish populations, certain Y-DNA haplogroups, markers passed down through the paternal line, are found at unusually high frequencies. The most significant of these are J1 and J2, which point directly to an ancestral homeland in the Levant. This means that Jewish men from vastly different backgrounds, whether Ashkenazi from Poland, Sephardi from Morocco, or Mizrahi from Iraq, often share paternal ancestry dating back over 3,000 years. Even more fascinating, studies have shown that the Y-DNA of Jewish populations has more in common with Middle Eastern groups like Lebanese, Druze, and Bedouins than with their European or North African neighbors. In other words, despite centuries of migration, Jewish DNA has preserved its ancient origins. But the story doesn't end there. Maternal DNA, passed through mitochondrial DNA, mtDNA, tells an equally intriguing story. While Ashkenazi Jewish men show clear Middle Eastern paternal ancestry, their maternal DNA suggests a different journey. A significant portion of Ashkenazi Jewish women can trace their lineage back to a small group of European founders who lived around 1,000 years ago. This suggests that while Jewish men migrated to Europe, they likely took local wives who later became part of the Jewish community. This unique blend of Middle Eastern paternal ancestry and European maternal ancestry helps explain why Ashkenazi Jews are genetically distinct from both Europeans and Middle Eastern populations. It also supports the idea that Jewish communities expanded through a combination of migration, conversion, and cultural adaptation, while still maintaining a core genetic heritage from their ancient Levantine ancestors. Sephardi and Mizrahi Jews, on the other hand, show a much stronger connection to the Middle East on both their maternal and paternal lines. Their mtDNA lineages often match those found in populations from the Near East, Mesopotamia, and North Africa, suggesting that their genetic continuity was preserved through endogamy, marrying within the community. But perhaps the most famous genetic discovery related to Jewish ancestry is the Cohen modal haplotype, a specific set of genetic markers found in the Y-DNA of Jewish men who claim descent from the ancient priestly class, the Kohanim. Remarkably, studies have confirmed that over 50% of self-identified Kohanim, whether Ashkenazi or Sephardi, share this unique genetic signature, suggesting that the priestly lineage has been passed down for thousands of years, just as Jewish tradition claims. So what does all of this tell us? It tells us that Jewish DNA carries an unbroken genetic legacy stretching back to the biblical era. It confirms what history and tradition have long suggested, that the Jewish people are linked by more than just religion and culture. They are connected by ancestry, by migration, and by a shared genetic thread that weaves through time. But the story of Jewish DNA isn't just about the past, it's also about the present and the future. As genetic research advances, new discoveries continue to reshape our understanding of Jewish origins, identity, and health. In the next sections, we'll explore how genetic bottlenecks shape the Jewish genome, why Ashkenazi Jews have such a high prevalence of inherited diseases, and what these findings mean for the future of genetic medicine. Imagine being part of a family that has been separated for thousands of years, scattered across continents, exposed to different cultures, yet still carrying the same ancestral DNA. That's exactly what happened with the Jewish people. From the ancient lands of Israel to the farthest corners of the world, Jewish communities have preserved a unique genetic signature despite centuries of migration, exile, and adaptation. But how did this happen? The Jewish diaspora began over 2,500 years ago, when the Babylonians destroyed the first temple in Jerusalem and exiled the Jewish people. Later, the Romans intensified this dispersion after the destruction of the Second Temple in 70 CE, scattering Jewish communities across Europe, North Africa, and the Middle East. Each of these groups settled in different regions, yet they maintained strong cultural and religious ties that influenced their genetics. Unlike many other migrating populations that intermarried extensively with locals, Jews often lived in tight-knit communities, practicing endogamy, marrying within their own groups. This created genetic bottlenecks, where a relatively small number of ancestors contributed to the gene pool of future generations, leading to distinct genetic patterns. For example, Ashkenazi Jews, who primarily settled in Central and Eastern Europe, experienced a significant founder event around 700 to 800 years ago. Genetic studies show that today's Ashkenazi Jews descend from a small population of a few hundred individuals, which is why they share certain genetic traits at a much higher frequency than the surrounding European populations. Similarly, Sephardi Jews, expelled from Spain and Portugal in the 15th century, carried their Middle Eastern and Iberian genetic markers as they resettled in the Ottoman Empire, North Africa, and the Americas. But the most fascinating aspect of Jewish genetics is how different Jewish groups, Ashkenazi, Sephardi, Mizrahi, and even smaller communities like the Bene Israel of India or the Beta Israel of Ethiopia, still share a core Middle Eastern genetic ancestry. Despite living among different host populations for centuries, Their DNA still traces back to the Levant, proving a deep-rooted genetic continuity that transcends geography. One of the most surprising genetic discoveries came from the Lemba people of southern Africa, who have oral traditions claiming Jewish ancestry. Genetic testing revealed that a significant portion of Lemba men carry Y-DNA markers commonly found in Jewish priestly lineages, suggesting a possible ancient connection. Similarly, studies have shown that Palestinian Arabs and Jews share close genetic ties, reinforcing the historical reality that they come from the same ancestral population before their paths diverged. The Jewish diaspora didn't just shape history, it left a permanent imprint on their DNA. Through migration, resilience, and cultural preservation, Jewish communities have retained a genetic signature that is both ancient and uniquely distinct. Picture a massive tree with thousands of branches, but all those branches stem from just a handful of roots. That's exactly how genetic bottlenecks work, and the Jewish people are one of the most striking examples of this phenomenon. Today, millions of Ashkenazi, Sephardi, and Mizrahi Jews share genetic similarities not because of widespread mixing, but because their ancestors came from a small, isolated group that expanded rapidly. But how did this happen? A genetic bottleneck occurs when a population is drastically reduced in size, leaving only a few individuals to pass on their genes to future generations. This leads to low genetic diversity, meaning many people inherit the same mutations and traits. Jewish populations, particularly the Ashkenazi Jews, went through multiple bottlenecks over the centuries, creating a distinct genetic identity. The most well-documented bottleneck among Ashkenazi Jews occurred around 700 to 800 years ago, when their population dropped to just a few hundred individuals. Despite growing into a community of millions, modern Ashkenazi Jews still carry the genetic imprint of those few founders. Studies have found that nearly half of Ashkenazi Jews descend from just 4 women, whose mitochondrial DNA continues to be passed down through generations. This bottleneck effect also played a role in the genetic makeup of Sephardi and Mizrahi Jews, though their histories are slightly different. The expulsion of Jews from Spain and Portugal in the late 15th century forced Sephardic Jews into smaller, more insular communities across the Ottoman Empire and North Africa. Mizrahi Jews, having lived in the Middle East for millennia, also experienced genetic bottlenecks due to persecution and forced migrations, though to a lesser degree than the Ashkenazim. But genetic bottlenecks don't just shape ancestry, they also affect health. Because Ashkenazi Jews descend from a small group, they have a higher prevalence of certain genetic diseases, like Tay-Sachs, Gaucher disease, and BRCA1-BRCA2 mutations linked to breast cancer. These mutations became more common simply because they were present in the small group of founders and were passed down through generations. This is why genetic testing is now a crucial part of medical care for Jewish communities, allowing individuals to screen for inherited conditions and make informed health decisions. What's truly fascinating is that despite living in different parts of the world for centuries, Jewish populations still share a core genetic heritage. A Sephardi Jew from Morocco, a Mizrahi Jew from Iraq, and an Ashkenazi Jew from Poland may look different on the surface, but their DNA tells a deeper story, one of resilience, survival, and shared ancestry. The Jewish genetic bottleneck is a testament to how history, migration, and survival shaped the very fabric of who we are. Even as Jewish communities expanded and adapted to new lands, their genetic legacy remains a link to a shared past that continues to define their present. Imagine tracing your family tree and discovering that nearly half of your entire lineage comes from just 4 women who lived nearly 1,000 years ago. Sounds impossible, right? Yet, this is exactly what genetic studies on Ashkenazi Jews have revealed. Unlike paternal ancestry, which follows the Y chromosome passed from father to son, maternal ancestry is traced through mitochondrial DNA, passed exclusively from mother to child. This unique genetic inheritance holds fascinating secrets about the Jewish diaspora. Among Ashkenazi Jews, mitochondrial DNA, mtDNA, studies have revealed an extraordinary pattern. Almost 40% of Ashkenazi Jews today descend from just 4 maternal ancestors who likely lived in Europe around a millennium ago. These 4 women contributed a significant portion of the modern Ashkenazi gene pool, and their genetic legacy continues in millions of people today. But where did these founding mothers come from? This question has sparked a heated debate among geneticists. Some studies suggest that these women were of Middle Eastern origin, indicating that early Jewish men who migrated to Europe married women from the same ancestral homeland. However, Other research argues that these women were local European converts, especially from regions like Italy, where Jewish communities thrived during the early medieval period. If true, this would mean that while Jewish paternal ancestry remained deeply Middle Eastern, the maternal lineage absorbed significant European influence. Sephardi and Mizrahi Jews, on the other hand, show less evidence of a strong maternal bottleneck. Instead, their mtDNA points to a broader range of Middle Eastern maternal ancestors, suggesting that these communities remained more genetically stable over time. For example, Iraqi and Persian Jews trace much of their maternal ancestry to ancient Near Eastern women, reinforcing their deep historical roots in the region. One of the most interesting cases is that of Ethiopian Jews, Beta Israel. Their mtDNA closely matches that of their non-Jewish Ethiopian neighbors, which supports the theory that the Beta Israel community descended from local converts who adopted Judaism centuries ago rather than migrants from the Levant. A similar pattern is observed among Indian Jewish communities like the Bene Israel, whose maternal ancestry shows strong South Asian influence, mixed with some Middle Eastern genetic markers. But why does maternal ancestry matter so much in Jewish identity? Unlike many cultures that trace lineage through the father, traditional Jewish law determines Jewish status through the mother. This means that for centuries, a child's identity as Jewish wasn't defined by their last name or their father's heritage, but by the mitochondrial DNA passed down through their mother. Whether this rule was influenced by ancient genetic realities or religious philosophy remains a mystery, but it has certainly reinforced the importance of maternal lineage in Jewish history. In the end, Jewish maternal ancestry is like an intricate mosaic. Some pieces point directly to the ancient Levant, while others reflect the regions where Jews settled over centuries. Yet, despite the diversity, the thread connecting all Jewish communities remains strong. Their maternal lineage is not just about genetics, it's a living record of migration, survival, and adaptation. Throughout history, the Jewish people have been known for their deep religious traditions, but what if science could confirm an unbroken bloodline dating back to biblical times? One of the most fascinating discoveries in genetics is the unique Y chromosome lineage of Jewish men, which not only connects them to their ancient Middle Eastern ancestors but also reveals an extraordinary genetic legacy among the priestly class, the Kohanim. The Y chromosome is passed unchanged from father to son, making it a powerful tool for tracing paternal ancestry. Unlike mitochondrial DNA, which reveals maternal origins, the Y DNA in Jewish men strongly links them to the ancient Levant, supporting historical accounts of a common Israelite ancestry. This means that whether a Jewish man comes from Europe, North Africa, or the Middle East, he is likely to share deep genetic ties with other Jewish men across the world. One of the most remarkable discoveries in Jewish Y-DNA research is the Cohen modal haplotype, CMH, a specific genetic signature found in a high percentage of Jewish men who identify as Kohanim, or descendants of Aaron, the brother of Moses. In Jewish tradition, the Kohanim are priestly caste who have served in religious roles for thousands of years. Could modern genetics prove that this lineage is real? Studies have shown that a significant proportion of Kohanim, regardless of where they live, carry nearly identical Y chromosome markers. This suggests that many Jewish priests today may indeed be descended from a single common ancestor who lived over 3,000 years ago, a timeline that aligns almost perfectly with the biblical account of the Exodus and the establishment of the Israelite priesthood. But the Y-DNA story doesn't end with the Kohanim. Jewish paternal ancestry also reveals fascinating connections to Middle Eastern and neighboring populations. Despite centuries of dispersion, Jewish men from different communities, Ashkenazi, Sephardi, Mizrahi, and even some African and Indian Jewish groups, share Y-DNA haplogroups that are commonly found in the Levant. This means that while Jewish communities may have absorbed some genetic influence from surrounding non-Jewish populations over time, their paternal lineage remains remarkably consistent and Middle Eastern in origin. However, there are some surprises hidden in the Jewish Y-DNA data. The Ashkenazi Levites, another hereditary group in Jewish tradition, show a completely different pattern. Instead of primarily Middle Eastern Y-DNA, over 50% of Ashkenazi Levites carry a European Y chromosome lineage, particularly from Eastern Europe. Some researchers believe this may indicate a non-Jewish founder event, perhaps a single European man who converted to Judaism and whose descendants went on to form a significant portion of the Levite class among Ashkenazi Jews. This has led to debates about whether this lineage came from the Khazars, a Turkic people who may have converted to Judaism in the early medieval period. though modern studies lean more toward a Near Eastern origin. Another fascinating case is the Lemba people of southern Africa, a Bantu-speaking group with oral traditions claiming Jewish ancestry. Genetic studies have found that a subset of Lemba men carry the Cohen modal haplotype, suggesting that at least some of their ancestors may have had Jewish or Semitic origins. Their Y-DNA also shows Middle Eastern markers, reinforcing their claims of descent from ancient Jewish traders or migrants. So, what does Jewish Y-DNA ultimately reveal? It confirms what historians and Jewish tradition have long maintained, that despite thousands of years of migration, separation, and adaptation, Jewish men from different parts of the world share a common paternal ancestry that traces back to the ancient Israelites. It also proves that Jewish identity isn't just cultural or religious, it's deeply embedded in their DNA, preserving a connection to the past that is both profound and unbroken. Imagine being able to trace your maternal lineage back over 1,000 years, to a small group of women whose genetic legacy still lives on in millions of people today. That's exactly what modern genetics has uncovered about Ashkenazi Jews. Unlike the paternal Y-DNA, which shows a strong connection to the Middle East, the maternal genetic history of Ashkenazi Jews is far more complex, and it has led to one of the biggest debates in Jewish genetic research. Mitochondrial DNA, or mtDNA, is passed exclusively from mother to child. This means it's the key to unlocking maternal ancestry, offering a window into the past that male-inherited DNA cannot. When scientists analyzed the mtDNA of Ashkenazi Jews, they discovered something astonishing. 40% of all Ashkenazi Jews descend from just 4 women who lived in Europe around 1,000 years ago. These women, known as the 4 founding mothers, played an extraordinary role in shaping the genetic makeup of millions of Ashkenazi Jews today. But here's where things get controversial. Were these 4 women originally from the Middle East, or were they European converts to Judaism? Some studies argue that their DNA shows distinct Near Eastern origins, suggesting that they were part of early Jewish migrations into Europe. Other research, however, points to European ancestry, meaning that Jewish men who migrated to Europe may have married and converted local women, leading to a significant maternal European influence. What makes this even more fascinating is that Sephardi and Mizrahi Jews do not show the same pattern. Unlike Ashkenazim, Jewish populations from North Africa, the Middle East, and Persia have maternal lineages that are overwhelmingly Middle Eastern, closely resembling other Semitic-speaking populations in the region. This suggests that while Jewish men largely maintained their Middle Eastern paternal ancestry, the maternal ancestry of Ashkenazi Jews may have been shaped by their time in Europe. The debate over these 4 women isn't just an academic question. It has major implications for Jewish history and identity. If they were originally Middle Eastern, it reinforces the idea that the Jewish people maintained their genetic continuity despite migration. But if they were European, it could mean that Jewish identity in Europe was more influenced by local conversions than previously thought. Interestingly, Jewish maternal DNA studies also reveal something else: bottleneck effects. Because Ashkenazi Jews descended from such a small number of maternal ancestors, their mtDNA has remained relatively homogeneous. This is why certain genetic disorders, like Tay-Sachs and certain breast cancer mutations, are more common in Ashkenazi Jews today. These conditions aren't just a result of inbreeding. They are genetic signatures of a small, isolated population that grew rapidly over centuries. But Ashkenazi Jews aren't the only ones with intriguing maternal DNA. The Beta Israel, Ethiopian Jews, show mtDNA patterns that closely match their non-Jewish Ethiopian neighbors, suggesting that their maternal ancestry comes from local populations rather than ancient Israel. This aligns with historical accounts that Beta Israel likely emerged from a mix of Jewish migrants and local converts. Similarly, Indian Jewish communities, like the Bene Israel and Koshin Jews, show a strong South Asian maternal lineage, while their paternal Y-DNA remains linked to the Middle East. Again, this suggests that Jewish men likely migrated to India and married local women, who then carried on Jewish traditions through the maternal line. Even more mysterious are the Jewish communities of the Caucasus, like the Mountain Jews and Krimchaks, whose mtDNA shows a mix of Middle Eastern and local ancestry, reflecting centuries of interactions with surrounding populations. So, what does all of this tell us? Unlike Jewish paternal ancestry, which remains overwhelmingly Middle Eastern, Jewish maternal ancestry tells a different story, one of migration, adaptation, and occasional assimilation. While some Jewish communities preserved their maternal Middle Eastern roots, others, particularly Ashkenazi Jews, seem to have absorbed new maternal lineages from their host regions, blending ancient traditions with new genetic influences. This research not only deepens our understanding of Jewish history, it also highlights the resilience of Jewish identity. No matter where Jewish communities migrated, no matter how their maternal or paternal ancestry shifted over time, their cultural and religious traditions remained intact, carried through the generations. Imagine carrying a hidden piece of history within your DNA, one that connects you to a small group of ancestors from centuries ago. For Ashkenazi Jews, this genetic legacy is more than just ancestry. It's the reason why certain diseases appear at higher rates in their population. This phenomenon is known as the founder effect, and it's one of the most fascinating aspects of Jewish genetics. The founder effect happens when a small group of people becomes isolated and their genes are passed down with little outside influence. Over generations, certain genetic traits, including disease-causing mutations, can become more common simply because they were present in the original founding population. In the case of Ashkenazi Jews, a small group of ancestors, perhaps just a few thousand people, formed the basis of the modern community. This genetic bottleneck means that Ashkenazi Jews today share more genetic similarities with one another than most other ethnic groups. One of the most well-known consequences of the founder effect is Tay-Sachs disease, a fatal neurodegenerative disorder that appears 100 times more frequently in Ashkenazi Jews than in the general population. This disease is caused by a mutation in the hexa gene, which leads to the gradual destruction of nerve cells in the brain and spinal cord. Historically, Tay-Sachs was often diagnosed in infants, who tragically did not survive past early childhood. But Tay-Sachs is just one of over 20 genetic conditions that are disproportionately found in Ashkenazi Jews. Gaucher disease, another inherited disorder, affects the body's ability to break down certain fatty substances, leading to enlarged organs, bone pain, and severe fatigue. Unlike Tay-Sachs, Gaucher disease can sometimes be managed with enzyme replacement therapy, allowing many patients to live relatively normal lives. Then there's BRCA1 and BRCA2, the genetic mutations linked to breast and ovarian cancer. While these mutations can be found in other populations, Ashkenazi Jewish women have a 1 in 40 chance of carrying them, compared to 1 in 400 in the general population. This dramatically increases the risk of developing cancer, which is why genetic testing has become so important in Jewish communities. Interestingly, some of these genetic mutations may have originally provided a survival advantage. Scientists have speculated that certain mutations linked to Gaucher disease might have helped protect against tuberculosis, while carriers of Tay-Sachs mutations might have had some resistance to other neurological conditions. This is similar to how sickle cell trait protects against malaria in certain African populations. The Sephardi and Mizrahi Jewish populations have their own unique genetic disorders, though they tend to be less well known. For example, familial Mediterranean fever, which causes periodic fevers and inflammation, appears more frequently in Jews from North Africa and the Middle East. Glucose 6-phosphate dehydrogenase, G6PD, deficiency, a condition that leads to red blood cell breakdown, is also more common in Sephardic Jews, possibly due to its link to malaria resistance. One of the most unique cases is the Lemba people of southern Africa, who claim Jewish ancestry and carry certain genetic markers linked to Jewish populations. They also have a higher frequency of G6PD deficiency, similar to Jews of Middle Eastern descent. This suggests that their genetic history might be more connected to the ancient Jewish world than previously thought. But while these genetic risks are significant, modern science has provided life-saving solutions. Thanks to genetic screening programs, Jewish communities have drastically reduced the number of babies born with Tay-Sachs and other severe genetic disorders. In fact, since widespread genetic screening began, Tay-Sachs births in Ashkenazi Jewish populations have dropped by over 90%. Today, many Jewish couples undergo carrier screening before having children, ensuring that they are not both carriers of the same disease-causing gene. This type of genetic testing has even started shaping Jewish matchmaking customs. In Orthodox Jewish communities, organizations like Dor Yeshorim provide anonymous genetic screening to young men and women before marriage. If two carriers of the same disease mutation are found to be a match, they are discouraged from getting married to prevent passing on the condition to their children. At the same time, genetic research on Jewish diseases is leading to breakthroughs that help all of humanity. The study of BRCA mutations in Ashkenazi Jews has helped doctors identify breast cancer risks in women of all backgrounds. Research on Gaucher disease led to enzyme replacement therapies that are now used to treat other lysosomal storage disorders. So, what does this all mean? The genetic legacy of Jewish populations is a double-edged sword. It carries the risks of inherited diseases, but also provides vital medical insights that benefit the world. And as genetic research advances, Jewish communities are using this knowledge to improve health, make informed choices, and even reshape traditions. Across centuries and continents, Jewish communities have developed distinct traditions, languages, and cultures. But what if, despite all these differences, their DNA tells a story of deep, unbroken connection? Science now confirms what history has long suggested. Jewish populations worldwide share a unique genetic bond that traces back to a common ancestral source. But how is this possible when Jews have lived in vastly different environments, from the deserts of Yemen to the frozen landscapes of Russia? Recent studies show that most Jewish communities share a significant portion of their genetic makeup with each other, particularly in their paternal Y-DNA. Whether it's Ashkenazi Jews from Eastern Europe, Sephardic Jews from Spain and North Africa, or Mizrahi Jews from the Middle East, the majority of their paternal lineages can be traced to the ancient Levant, the homeland of the Israelites. This genetic link remains strong, even among communities that were separated for over 1,000 years. Interestingly, the maternal genetic picture is more complex. While some Jewish maternal lines also originate in the Middle East, others show signs of local European and North African ancestry. This suggests that Jewish men, throughout history, often married local women who then became part of the Jewish community through conversion. However, despite this mixture, genetic drift and endogamy, the practice of marrying within the community, help preserve the unique Jewish genetic signature. One of the most fascinating aspects of Jewish genetics is how closely related Jewish groups remain despite geographic separation. For example, genetic studies reveal that Ashkenazi Jews are more closely related to Sephardic Jews than they are to their non-Jewish European neighbors. Similarly, Yemenite Jews, who lived in near isolation for centuries, still share strong genetic ties with Jews from Iraq and Iran. Even the Jewish communities of India and Ethiopia, which have historically been considered outliers, show significant ancestral links to other Jewish groups. This genetic connection is not just a scientific curiosity. It reinforces a shared identity that has survived exile, migration, and persecution. It also explains why Jewish populations have higher frequencies of certain hereditary traits and diseases, as genetic isolation led to the preservation of specific mutations. But more than that, it proves that despite being scattered across the world, Jewish communities remain, quite literally, family. For years, a controversial theory has suggested that Ashkenazi Jews are not of Middle Eastern origin but instead descend from the medieval Khazars, a Turkic people who supposedly converted to Judaism in the 8th or 9th century. But what if this theory, despite its popularity in some circles, has no scientific foundation? Modern genetic studies have now provided a clear answer, and it completely dismantles the Khazar hypothesis. The Khazar theory suggests that a significant portion of Ashkenazi Jews originates from a mass conversion of Khazars in what is now Russia and Ukraine. According to this claim, instead of descending from ancient Israelites, Ashkenazi Jews would be largely European or Central Asian in ancestry. However, when scientists examined the DNA of Ashkenazi Jews, the evidence told a very different story. Multiple genetic studies, including those by Beho et al., Nebel et al., and Hammer et al., have confirmed that Ashkenazi Jews share the vast majority of their paternal ancestry with Sephardic and Mizrahi Jews, as well as with Middle Eastern populations like Palestinians, Druze, and Bedouins. Their Y-DNA haplogroups, such as J1 and J2, are characteristic of populations from the Levant, not Central Asia. If the Khazar theory were true, Ashkenazi Jewish paternal DNA should resemble that of Turkic or Mongolic groups, but it does not. Even on the maternal side, the evidence contradicts the Khazar hypothesis. While Ashkenazi Jewish maternal ancestry is more diverse, showing some European and Middle Eastern influence, it still lacks a clear connection to Central Asian or Turkic populations. If large numbers of Khazar women had converted to Judaism and formed the backbone of Ashkenazi maternal ancestry, we would expect to see significant Central Asian genetic markers in Ashkenazi mitochondrial DNA. Yet, no such markers exist. Furthermore, whole genome studies place Ashkenazi Jews genetically between Middle Eastern and Southern European populations. This suggests that while some European admixture did occur, likely through marriage with local European women during the early centuries of Jewish settlement in Europe, the core ancestry remains firmly rooted in the ancient Near East. If Ashkenazi Jews were primarily Khazars, their genome would be vastly different from other Jewish populations, but instead, they share a strong genetic connection with Sephardic, Mizrahi, and even some North African Jewish groups. So why does the Khazar theory persist? Some historians believe it gained traction for political reasons, while others see it as an attempt to undermine Jewish claims to Middle Eastern ancestry. However, the overwhelming genetic evidence leaves little doubt. Ashkenazi Jews, like their Sephardic and Mizrahi counterparts, are primarily descended from the ancient Israelites. The Khazar theory, once a speculative historical narrative, has been thoroughly debunked by science. For centuries, Jewish genetic heritage has been shaped by migration, isolation, and historical upheavals. But today, with the rise of modern genetics, DNA testing, and global intermarriage, what does the future hold for Jewish genetics? Will the distinct genetic markers that have defined Jewish ancestry for millennia remain? Or are we witnessing a new chapter in Jewish genetic history? One of the biggest factors influencing Jewish genetics today is intermarriage. In many Western countries, more than half of Jewish individuals marry non-Jews. This trend increases genetic diversity, reducing the likelihood of inherited disorders common in endogamous Jewish communities. However, it also means that over time, the distinct genetic signatures that have characterized Jewish populations for centuries may become less pronounced. At the same time, genetic research is advancing at an unprecedented rate. Scientists are now able to trace Jewish ancestry with incredible accuracy, confirming connections between Jewish groups and their Middle Eastern roots. Large-scale DNA databases, like those used in genealogy testing, are helping Jews reconnect with lost relatives and discover hidden Jewish ancestry in families that may have lost touch with their heritage due to forced conversions or historical migrations. Medical genetics is another area where Jewish DNA research is making a major impact. Studies on Ashkenazi genetic diseases, for example, have led to breakthroughs in understanding conditions like breast cancer, BRCA mutations, Parkinson's disease, and Crohn's disease. These discoveries are now benefiting the wider medical community, as researchers apply their findings to non-Jewish populations who share similar genetic traits. But there's also a growing ethical debate. Some fear that genetic research could be misused to reinforce divisive narratives about ethnicity and identity. Others worry about the potential for genetic discrimination, where insurance companies or governments might use DNA data to make unfair policies. The Jewish community has a long history of facing persecution based on ancestry. Could genetic research be weaponized in the wrong hands? Despite these concerns, the future of Jewish genetics is filled with possibilities. The combination of historical DNA analysis, medical advancements, and genealogical research is creating an unprecedented opportunity for Jews worldwide to explore their ancestry, improve healthcare, and preserve their heritage in a rapidly changing world. One thing is certain: Jewish DNA is not just a relic of the past, it's an evolving story, one that continues to unfold with every generation.